The same thing is going on. So thymine to adenine, adenine to thymine, guanine to cytosine. Same thing. Base pairing. Even though I don't draw very well, it's looking messy, but you can do it on your own papers. But whatever your nucleotides are, do the base pairing. So thymine to adenine, adenine to thymine, guanine to cytosine. And once again, ligase will sew them together into one strand. So yes, the same thing exactly is occurring down below and on top. Down below and on top. But you can see the composite nature of the polymerase because one part of it is separating the strands, one part is going after other nucleotides, one part is sewing together the new ones into one strand, and all of that is DNA polymerase. They are now.
No, I mean,
It's not a covalent or ionic bond. Hydrogen bond means the charges are kind of attracting each other, but it's not a direct bond, no. So it is yes and no.
but it's not like one can go broke.
Correct. So that was just for this early part, but the process keeps going. In other words, the DNA keeps unraveling. So we keep on adding on more nucleotides, the helicase, the ligase, the polymerase. Everything keeps working. It's going to go the entire length of the DNA. The entire DNA is going to come apart. Now again, it's not all at one time. It just is being pulled apart. So you keep pulling it apart, you keep on adding on the new stuff. Eventually, you do get to the end. Eventually, you'll get to the end of the DNA, and the DNA will literally come apart. It will separate. So when that happens, we're going to have two separate molecules. Making new DNA. Replications making new DNA. So you can see how the new ones are being made here. And again, this just keeps on separating. So you keep on doing this until you get to the end. Then you have this and this. Now something to draw your attention to. If you look at each of these, you can see the strand up here is the old strand or the original DNA strand. And this one here is the new one. The new one that was forming. Same thing down here. This is the original old strand. And this is the new one that was forming. So if we continue that thought process up here, each of these DNAs are going to be half and half. Old, new, new, old. Each of those DNA molecules is going to be one old strand and one new strand. And this is called semi-conservative replication. semi-conservative, the semi, which as you know means half, the semi-conservative is referring to the fact that each DNA molecule is half and half. Half old, half new. That's your semi-conservative. Now, that may or may not still be clear, so let me give you a counterexample. If we had conservative replication, which we do not, I do not know any example in nature of conservative replication. As far as I know, it does not occur. But if we did have conservative replication, if it did exist, it would look like this. So you still have two DNA molecules, you're still replicating. But now, one molecule will be your original molecule. You've got two old strands there. That's your original DNA. And then the other molecule will be a brand new molecule with two new strands. See, that would be conservative replication. It's not half and half. It's either old or it's new. Now again, to my knowledge, that does not exist in nature. I know we've seen all quarter, there's a million different exceptions, a million variations, and maybe somebody does do this, but I don't think so. But that does not mean it does not exist. Because actually this does exist in everyday human activity. Think of some activity that you do all the time. Now, it may not be every day, maybe I'm exaggerating. But certainly several times a week. I'd be very surprised if you don't do something like this several times a week. Can you think of anything that would qualify? Pardon me?
Which one?
Conservative replication. So again, it does not occur in nature, but I'm saying it does occur in more or less day-to-day life. And you think of anything that you do, probably you did it today even, somewhere. Maybe not, but several times a week.
Taste buds?
No, not biological, remember. Not biological. I'm talking human activity of some sort. But not brush your teeth. You're not making new teeth. Yeah.
Oh, I see what you're saying.
I'm talking about conservative replication. Pardon me? Taking notes? So you're taking notes double?
I mean, you're taking notes,
Well, that, actually, you might be right. I didn't have it true. No, what I had in mind is photocopying. Photocopy, Xeroxing. So you put the paper into the machine. When the paper comes out, you get the original paper back. And of course, you have a brand new copy. See, that would actually be conservative replication. It's not biological. But we do it all the time. So it may not be every day, but again, I would be surprised if you don't do it several times a week. So that's an example. So you're starting out with the original, you get back the original, and you also get a brand new copy. But this does not exist inside of our blood cells. So semi-conservative, and that's really what exists. So we go through this whole thing, and we end up with one molecule, half old, half new. Half old, half new. Any questions on that? The more questions you ask, the less new stuff I get you. Okay. I knew that would work.
I don't understand how it would end up like that.
This, well, you see, it really does not. Remember, replication, that is not heredity. Replication really is just involved with making new cells and making the DNA for the new cells. But reproduction involves new organisms, making children and everything. And then heredity involves giving DNA to our children. And of course, we got it from our parents, and so on and so on. So that involves not replication, but transcription. Now, to explain what that is, let me ask you something we talked about a long time ago, and now we're coming back to it. Does anybody remember what we talked about a while ago? What is the actual function of DNA? Does anybody remember? Don't say heredity. We know that, but blueprint for what?
Making proteins.
Making proteins. You're right, you didn't finish it. So making proteins. So the function of DNA is to make proteins. Now, of course, as you know, the DNA itself is not making proteins. Ribosomes are making proteins. The DNA, as she said, is actually making the blueprint, it's giving the instructions to make the proteins. But there's a basic flaw. The DNA is in the nucleus and it never leaves. The ribosome is in the cytoplasm and it never leaves. So there's got to be some way of transporting or transmitting or something the DNA instructions into the cytoplasm to the ribosome. Well, the method for doing that, and I'm sure most of you know it already, is RNA. Transcription is the synthesis of RNA. Everybody has RNA. Everybody has RNA. I know that. Now, she's asking a very good question. I'll rephrase it. Her question's a good one. I'm saying that RNA is needed in order to go from DNA to the ribosomes and so on. So, and the DNA is in the nucleus. Well, of course, bacteria don't have a nucleus. So the DNA for the bacteria is just in the cytoplasm. So in that sense, would it be necessary for the bacteria to have RNA? Yes, it still is. The fact is, you still need something to go from, bacteria actually have one single chromosome. So you need something to go from that one chromosome to the ribosome. The ribosome apparently in the bacterial cell does not go to the chromosome. So you still need a messenger. So a good question, but in actuality, the bacteria still needs the RNA. And of course, ultimately, we get that from the bacteria. So we need to make RNA. We need to talk about that because we haven't, of course, up until right now. DNA and RNA. Obviously, one difference is the sugar. DNA has deoxyribose. RNA has ribose. And by the way, before I forget to mention it, RNA has nucleotides just like DNA does. They both have nucleotides. It's the same thing, sugar, base, phosphate. So they both have nucleotides. Okay, so for the RNA, it's got ribose. Second difference, DNA, of course, is a double-stranded molecule. So DS, double-stranded. RNA is single-stranded. So RNA just has one strand. And then the final difference, so you still got the bases, 4 bases. So for DNA, adenine, guanine, cytosine, thymine. For RNA, adenine, guanine, cytosine, and now a different 4th base. Uracil. Uracil. uracil basically takes the place of thymine. So in RNA, wherever you expect to see thymine, you have uracil. So these are the three differences. The sugars are different, double-stranded versus single-stranded, and essentially thymine or uracil. The other three bases are the same. Any questions on that before I erase it? Is everybody okay? Transcription, we're going to make RNA, and by the way, I might as well finish this off. Heredity actually has two parts. Transcription is the first part. Translation is the second part. Synthesis of protein.
Transcription is the first part.
Yeah, both of those are heredity together. So we're doing transcription right now. We're going to make the RNA first. So for this one, we need a similar enzyme as for replication, but this is called RNA polymerase. RNA polymerase. Now it sounds just like DNA polymerase, which might be confusing, but actually it's not bad, because the name itself tells you what you're making. DNA polymerase makes DNA. RNA polymerase makes RNA. So even though it sounds just alike, but actually the names will tell you what's going on. So in this case, we want RNA polymerase because it's making RNA. And this is very, very similar to the DNA polymerase, meaning it's another composite enzyme. It's got the same things as before. It's got ligase again, helicase. There are a couple of differences, which I'll show you in a minute, but essentially very, very similar. So ligase, helicase, composite enzyme.
And this is transcription, right?
Yeah, all of this now is transcription.
And just to clarify, composite enzyme is the polymerase, which includes ligase, helicase,
Yes.
and various other.
Yes. Yeah. All of those together make up the polymerase. Yeah. So again, similar in nature to the DNA polymerase, but there are a couple of differences which you'll see real quick. So once again, while helicase, I need to erase this just to make